We're still talking about cell biology, and we've already discussed the major organelles. So in this video, we're going to focus on the cytoskeleton. The cytoskeleton is the internal framework of a cell that gives it its specific shape and structure and orientation in space, depending on the function of the cell. There are several basic types of filaments that make up the cytoskeleton. There are microfilaments, which are the thinnest filaments. There are microtubules, which despite also being called micro, are actually the thickest type of filament. And then in between the microtubules and the microfilaments are the intermediate filaments, which have a diameter about half as large as the microtubules. And there's myosin, which is a little bit larger than the intermediate filaments. So again, going from the thinnest to the thickest, it's microfilaments, intermediate filaments, myosin, and microtubules. Now, microfilaments are made of polymers of a protein called actin. Actin is really important for cellular motility. Whether you're talking about neutrophil chemotaxis or the motility of macrophages, or even the microvilli of the intestinal brush border, or activated platelets that change shape to plug up a bleed. All those kinds of cellular movements are mediated by the growth and contraction of actin microfilaments. Now, myosin works right alongside actin to generate muscle contraction. So muscle cells have lots of myosin filaments. Remember, myosin, the myo means muscle. Myosin was first discovered in muscle cells, but there's actually an enormous family of myosin proteins, which are mostly involved in cellular motility in one form or another. And muscle contraction is certainly a big part of that. For intermediate filaments, take a look in your study guide. There are a whole bunch of different types of intermediate filaments, which are found in specific types of cells and tissues, and you can identify different tissues by staining for these different filaments. This really isn't all that clinically relevant unless you're going into pathology, but this is important for you to know for step one. So the first one in your study guide is cytokeratin. Cytokeratin is a very strong filament found in epithelial cells. Cytokeratin contains the protein keratin, which is found abundantly in the skin. Cytokeratin is found in desmosomes and also in hemidesmosomes, which are attachments between epithelial cells and other epithelial cells, and then between the epithelial cells and the basement membrane. That's going to be covered more in the dermatology videos. Now, we're going to come back and fill in the far right column about stains in just a second, but the next row is desmin. Now, desmin is found in all types of muscle cells, smooth muscle cells, skeletal muscle cells, even cardiac muscle cells. The next one is GFAP, or glial fibrillary acid proteins, which are found in glial cells, such as astrocytes and Schwann cells and so on. So this is a type of ectodermal intermediate filament. Then there are the neurofilaments, which can be categorized by their molecular weight. So there's low, medium, and high molecular weight neurofilaments. And they're found in neurons, as you might guess, but specifically they're found in the axons of the neurons. There are also nuclear lamins, A, B, and C. Now, these are definitely important for you to know for your exam. I mentioned previously that nuclear lamins are like the skeleton of the nucleus. They provide the structural framework for the nuclear envelope. And we talked about how the cyclin B CDK1 complex was associated with the breakdown of the nuclear envelope and the nuclear lamins, which has to take place during mitosis in order for the nucleus to divide. The other thing worth knowing about nuclear lamins is that the mutations in the nuclear lamin genes are implicated in several diseases, including certain types of muscular dystrophy and also the disease progeria. Progeria is a disease that causes advanced cellular aging, so that a 5-year-old child might have wrinkles in the facial structure of an 80-year-old because all these nuclear lamins are mutated. And then the last one here is vimentin. Vimentin is an important structural component of connective tissue, including fibroblasts and endothelium. It provides a strong support for cellular membranes, and it keeps organelles like the nucleus and the endoplasmic reticulum and the mitochondria fixed within the cytoplasm. Now let's go back and fill in the far right column of this table, and we'll talk about how different immunohistochemical stains can be used to identify tumors based on the intermediate filaments that they contain. Again, different tissues have unique intermediate filaments. So if you had a metastatic tumor somewhere in the brain or the liver or the lungs or wherever, you do a biopsy, and then the pathologist would stain that tissue to try to identify which kinds of intermediate filaments were present in the tumor. That's going to help you determine what kind of tissue the primary tumor came from. Sometimes it's hard to identify where metastatic tumors originated from, and these stains are going to help you identify what the origin of the tumor is. Cytokeratin stain can be used to identify carcinoma, which is a cancer that arises from epithelial tissue. Desmin is found in muscle cells, we said. So if you have a sarcoma of the muscle cells, a myosarcoma, you could use a Desmin stain to help you identify that tumor. So this might be like a rhabdomyosarcoma or a leiomyosarcoma. GFAP is a helpful stain in identifying certain types of astrocytomas, such as a glioblastoma. And then a neurofilament stain stains neurons rather than glia. Now this can be a useful stain to help you identify a tumor of neural origin, such as a neuroblastoma, which is a childhood tumor that arises from sympathetic ganglion cells in the adrenal medulla. And then again, Vimentin is found in connective tissue. So that stain is mainly helpful in identifying sarcomas and also some carcinomas. But primarily I want you to associate Vimentin with sarcoma, and you can associate cytokeratin with carcinomas. So hopefully these aren't too hard, but chances are you're just going to have to sit down and memorize these. The largest type of cytoskeletal filament are the microtubules. Microtubules are tubes made up of proteins called tubulin. And there are two types of tubulin, alpha tubulin and beta tubulin. And first they come together to form a dimer, and then the dimers join together lengthwise to form something called a protofilament. And then those protofilaments come together side by side to form a cylindrical microtubule with a hollow core. And the way the alpha and beta subunits are arranged, they form a spiral or a helix that runs the length of the microtubule. Now in terms of function, I want you to think movement. Microtubules are involved with movement, a couple of different kinds of movement. For starters, microtubules are an important part of the structure of flagella and cilia, and they're what enable the cilia to move, as we're going to see shortly. Microtubules are also part of the mitotic spindles, which are what pulls the duplicated chromosomes apart during nuclear division. Now, this is a simplified explanation, but basically during mitosis, the microtubules assemble toward the chromosomes and latch on, then they disassemble very rapidly and pull those chromosomes apart so the cell can divide. Microtubules polymerize and grow very slowly, but they can collapse fairly quickly. So if something prevents the microtubules from growing and then collapsing, that'll interrupt mitosis. So what drugs act on microtubules? Well, the first group are cancer drugs, and there are two groups of cancer drugs that act on microtubules: vinca alkaloids and taxanes. The vinca alkaloids are vincristine and vinblastine, and they work by blocking the polymerization of microtubules. They prevent the microtubules from growing. And we're going to talk about that at the very end of the course in the oncology section. And then the taxanes, or taxols, include drugs like paclitaxel and docetaxel, which work by hyperstabilizing the microtubules. So the microtubules can grow, but then they can't break down in order to pull those chromosomes apart. So the vincas and the taxols are similar in that they both interfere with microtubule function and they prevent mitosis in cancer cells, but on a cellular level, they actually work in very different ways. Then, other than the cancer drugs, there are the benzimidazoles, which are antihelminthic drugs like mebendazole, albendazole, thiabendazole. These are used to treat certain worm infections, hookworm, tapeworm, that kind of thing. You can remember that worms are bendy, so you're going to use mebendazole and albendazole to kill them. Griseofulvin is an antifungal drug that works on microtubules. It's mostly used for ringworm and nail fungus. There's also colchicine, which is an anti-inflammatory drug used to treat gout. So phagocytes use microtubules to move, and colchicine helps to reduce inflammation because the phagocytes can't move to where they need to go because they're using those microtubules to move and the drug is interfering with the microtubules. Now, one other function of microtubules is to allow the cell to move things around within the cytoplasm. Remember, microtubules are involved with movement. So they help cilia move, they help phagocytes move, they help chromosomes move during mitosis, and now they're helping the cell actually sort of ship things around from one part of the cell to the other. For instance, some cells are really big. Take a motor neuron for your big toe. The cell body is way up in the anterior horn cells of the L4, L5 region of the spinal cord, but the axon stretches all the way down to the big toe. That could be 3 or 4 feet long, depending on how tall you are. So there needs to be a mechanism to move things reliably from the cell body to the other end of the cell. So this process is called slow axoplasmic transport, and it basically uses the microtubules like a railroad track to guide things to the right location inside the cell. It's not just neurons. Other cells use microtubule tracks to move things around. So how exactly does this work? Well, the railroad cars that actually move things up and down these microtubule tracks are called molecular motor proteins. And there are 2 of these molecular motor proteins that you should know about, dynein and kinesin. Microtubules are oriented with a plus end and a minus end. The end of the microtubule made up of alpha tubulin is the minus end, and the end with beta tubulins is the plus end. So dynein and kinesin attach to the outside of the microtubule, and they also attach to whatever cargo they're hauling, and then they walk along the microtubule from one end to the other, dragging their cargo with them. So kinesin walks toward the plus end of the microtubule, which is generally going to be away from the nucleus, so that's called anterograde transport. And then dynein walks in the other direction, toward the minus end, which is called retrograde transport. We're going to come back to dynein in just a second. Let's also talk about cilia and flagella, because they're both composed of microtubules. The flagella of sperm are structurally very similar to a cilium, just a lot longer. In motile cilia and flagella, the microtubules come together in a 9+2 arrangement. A circle of 9 pairs, or doublets, of microtubules on the outside, and then 2 individual microtubules running down the center. And then lots of dynein motor proteins stick out from the microtubules and walk along adjacent doublets, which allows each microtubule doublet to move relative to another doublet, so that the cilium moves as a unit. Now, where in the body do we find cilia? We find them in the respiratory tract, including the trachea and the bronchioles, and also the paranasal sinuses, because cilia help to sweep out mucus and bacteria and debris. The fallopian tubes are ciliated, because the cilia help to sweep the ovum, or a fertilized zygote, down toward the uterus. So just keep that list of ciliated structures in your mind for just a moment. Then one other thing to know about how ciliated cells work is how they coordinate their ciliary motions. If each ciliated cell was doing its own thing, you wouldn't really be able to sweep anything along in a particular direction. You'd just kind of be going randomly. But instead, what we actually see is a coordinated ciliary contraction taking place that moves things in one direction. So how does one ciliated cell communicate with another ciliated cell to coordinate the contraction and create a wave-like motion? Well, it's accomplished by gap junctions between the cells. So gap junctions are important for the coordinated contraction of ciliated cells. That's just a tiny detail that's worth knowing for your test. Now, what happens if the molecular motor proteins in the cilia don't work? Well, there's a condition called primary ciliary dyskinesia, where the dynein doesn't work, and that leads to a lack of movement of the cilia in the respiratory tree, and in the sinuses, and in the fallopian tube, and even in the flagellum of the sperm. So in primary ciliary dyskinesia, there are immotile cilia due to a defect in the dynein arms. If the dynein can't walk like it's supposed to, you're not going to have movement of the cilia. So this results in infertility in both males and females, because in the males, the sperm are immotile, and in females, because the fallopian tubes aren't going to be able to sweep the zygote or the oocyte anywhere. There's also bronchiectasis, because these patients can't clear mucus out of the bronchial tree. There's chronic sinusitis, because they can't clear out the sinuses. And then cilia are also involved in determining the position of internal organs during embryological development. So about 50% of patients with primary ciliary dyskinesia also have situs inversus, where the organs are on the opposite side of the body. So the liver would be on the left and the spleen would be on the right. There's also dextrocardia, where the heart is over on the right. Now, not all patients with situs inversus have primary ciliary dyskinesia, and not all patients with primary ciliary dyskinesia have situs inversus. Now, if you have the triad of situs inversus, bronchiectasis, and chronic sinusitis, that's called Cartaginer syndrome. But again, about 50% of patients who have primary ciliary dyskinesia do not have situs inversus. So Cartaginer syndrome does not equal primary ciliary dyskinesia. When the cilia don't work, there's a 50-50 chance of whether your organs are going to be on the right side or the wrong side. So that's it for this video. Go ahead and answer the questions in the end of session quiz, and then we'll go over the answers together. All right, first question. What drugs act on microtubules? Well, I mentioned 4 cancer drugs: vincristine, vinblastine, paclitaxel, and docetaxel. Then there are a few others: benzimidazole antihelminthic drugs, mebendazole, albendazole, and thiabendazole. There's griseofulvin, which is an antifungal, and there's colchicine, which is an anti-inflammatory that acts on the microtubules in phagocytes. Next, what are the intermediate filaments found in the following tissue types and cellular structures? So in connective tissue, you see vimentin. In muscle tissue, you see desmin. In epithelial tissue, you see cytokeratin. And in the axons, you find the neurofilaments. And the last question, what are the defects seen in Cartaginer syndrome? So Cartaginer syndrome is a triad of bronchiectasis, chronic sinusitis, and situs inversus. And then because Cartaginer syndrome can also be a form of primary ciliary dyskinesia, you can also add infertility to that list, because infertility is a major feature of that disorder. So that's it for Foundations 4. I'll see you next time.
Two drugs with mechanisms not the same. The vincas and the taxanes are their names. One inhibits, the other a microtubule enhancer. Yet both are used to fight the dreaded cancer. As our tragic story doth begin, starring these inhibitors of tubulin, a pair of star-crossed lovers form our scene. Brave Paclitaxel and his Vin Christine. Soft! What light through yonder window breaks? It is the east, and Vin Christine is the sun. Arise, fair Vinca alkaloid, my sweet. Oh, that I were a Ewing sarcoma, and that thou wouldst inhibit polymerization of my microtubules and disrupt my mitotic spindles.
Oh, Paclitaxel, Paclitaxel, wherefore art thou Paclitaxel? Oh, what's in a name? That which we call a taxane by any other name would smell as sweet.
Why must our families oppose one another this way?
It is our very nature, my love. We Vincas inhibit cell division by inhibiting microtubules, while you taxanes stabilize microtubules.
Yes, but the end result is the same. Oh, why must this keep us apart?
Besides, you taxanes cause me such devastating hypersensitivity reactions.
Premedicate! Premedicate, my queen! With a steroid and an antihistamine!
Ronnie, this isn't working.
No, you're doing great! This show must go on!
This is a terrible way to study. I don't have time for these stupid role-playing games of yours. Can't believe I put on a corset for this.